recording. So today and the next lecture, we'll talk about the Deutorostomes. Deutorostomes are the creatures which developing, they have a very specific. I ask you if you want to use lockdown or not. So nobody responded, so I presume everybody's happy with the lockdown. Just kidding. Well, let me know. That's why we have a chat box, and I can see that you guys are kind of acting and hearing what I'm saying. So let me know which one you want to go. In any case, so Deutorostomes are creatures which have a very specific embryonic development. Remember, they have a radial cleavage. Whenever the gastrula start collapsing, that particular opening known as the blastopore will turn into anus. So basically, they start the digestive system from the back to the front. So in this case, all right, I got you. Thank you. So in this particular case, it's all about the embryonic development. And because of the embryonic development, they will have quite a few drastic differences from the classes or from the phyla we looked before. And of course, one of the largest one known as the vertebrates, and we'll hopefully start on vertebrates today, but there is a whole bunch of, well, I should say there is a couple of phylum which are considered to be invertebrate. Why not? Well, because they don't have a vertebral column. Even though some of them are extremely close related to us, they still don't have a vertebral column. Therefore, they are invertebrates. And this is what the majority of today's lecture will be. Invertebrates, but deuterostomes. Again, deuterostomes, it's a very specific embryonic development. So, all right, so let's kind of take a look which groups we have. Echinoderms. Echinoderms, echinos means needle. Dermis means skin. Skin filled up with the needles. If you watch the videos, they already talk about the echinoderms quite nicely, the starfish, etc. If you haven't, once again, I'm asking you, please do that particular video. Watch the video. It shows how the sea star turns into, well, not turns, but how the evolution worked. That the sea star flattened one, then goes into a round one, creating sea urchin, and it's extended, and that would be a sea cucumber, and so on and so forth. So here we're talking about the echinoderms, and I think next lab number 14, relatively short lab, we'll talk about the dissection of the sea stars. Hemichordates, well, the diversity as well as the diversity of the echinoderms and the sea star. So hemichordate, hemi kind of halfway. They're not exactly the, they don't have a notochord, which is kind of flexible along the body, but they do have some part of it. I'm not going too deep into that. We will not have much of the hemichordates in the lab. And then finally, chordates. Chordates are the creatures we belong to, that particular group, that particular branch, that particular flexible rod. It's initial internal skeleton. Later on, it will be substituted for the vertebral column. So me, as an adult, don't have, or neither of you, don't have actual that particular notochord. But our distant relatives, lanceolets, for example, they do have that particular notochord, and they don't have a vertebral column, so they consider to be invertebrate. All right, so first, basically, when you're looking at the echinoderms, doesn't look sea star, do not look very much as us. However, we are put at the same group because, again, we're looking at the characteristics within the development, within how the zygote develops into the adult organism or into the embryo. And again, we're talking about radial cleavage. Xylem develops from the ancient or primitive gut, and formation of the mouth at the end of the embryo. So we have the embryo start collapsing. That particular initial opening, the blastopore will turn literally into anus, and then it will continue to grow. And the secondary opening, whenever the tube will reach the opposite pole of the ball of the gastrula, that will become mouth. So mouth second, Deutorostome. This is what the term actually means. So some of the characteristics of the echinoderms, again, as the name suggesting, they have a spiny skin or filled up with needles. So we'll talk about that. They have a very unique, something which known as a water vascular system. This is the way how they slowly moving. They're not fast creatures, but they are very, very effective. As a matter of fact, in the video, they shows how effective vascular system is against the muscles of the mollusk. With the help of the tube feet, they will be able to pry open the, I think it's a clam or maybe a mussel shell, and then digest it, kill it, and eat it. So they do have endoskeleton, as a matter of fact. That's important part, which actually puts us closer to the echinoderms, let's say insects. Insects, of course, have they are protostomes, and they have an external skeleton. Larvae is bilateral. That's a very important point. And some of the scientists believe that at some point of time, a long time ago, about 1 billion years or so, one of those larvae, which didn't go through the symmetry of radial symmetry or going into the complete metamorphosis into the adult organism, start reproducing, and this is what lead to the group which we know nowadays as the Chordates, and later on Vertebrates. But we'll discuss that a little bit. Larvae is very similar to a simple chordate organism, bilateral symmetry, free swimming, and so on. Adults will have a pentaradial symmetry. Remember, as soon as we start talking about radial symmetry, there is no front end, which is going into and sensing the environment. There is no head. No head means that all of the sensors are spread around the body. If all of the sensors more or less equally, if all of the sensors more or less equally spread around the body, there is no concentration of the nervous tissue. No concentration of the nervous tissue, meaning there is no brain per se. There is something which is known as a nerve ring, but there is no concentration, no brain, so they are completely brainless, very successful group. Relatively slow creatures, still walking around, but not very fast. Well, slow, but very predators. That's the sea stars. So anyway, sea stars are, as well as many of other echinoderms, slow moving, sometimes sessile, means that they sit on the bottom of the ocean and wave their arms and grab whatever they can, and they just send it down into the stomach, into the mouth. From the mouth it gets into the stomach, digest it, and then come out of the anus. We'll talk about that. I'll show you the either or sides. Bumpy or spiny skin and covers with the interlocking calcareous plates. Within those particular interlocking calcareous plates, they have a very specific muscles, and because of that, the sea star able to extend or move its own arms. But the actual movement goes through the tube feet. I would highly recommend you to watch that particular video on echinoderms to figure it out, how the water vascular system works. I will explain it briefly using my hands, literally, and but it's much easier to see it in the animations. They have it in the video, and that's the reason it's a short videos, relatively nice videos comparatively. So unique, very unique things known as the water vascular system. Water vascular system is literally using water. It's a hydraulic system using a special channels throughout the body of the sea star. So everything starts from right here. From the top, on top of the sea star, and off center, you'll find that particular calcareous plate. Calcareous plate known as a sieve plate or madreporite. It's the same thing. Madreporite, okay? So the water literally from the environment goes into that particular sieve plate / madreporite. From there will be something which is known as a stone channel. From the stone channel, it goes into a ring channel, and the ring or ring canal. And the ring canal will be separated into, well, it will send the water into each one of those, something which is known as a radial canal or radial channels. So water from the madreporite goes into a stone canal, into a ring canal, into a radial canal. Okay, so that's how the water initially moves. Now, radial canals will be attached to a tiny little bitty bulbs. All right, those particular bulbs known as ampoules. So water filling out the ampulla, and the ampulla on the bottom attached to the tube feet. It's literally a tube foot. So guess what will happen whenever the ampulla contracts? Whenever the ampulla contracts, it's sending water down. As a result, the tube foot is extending, increasing, pushing it with the water. All right? Makes sense? And whenever the ampulla relaxes, water goes back, the tube foot is slowly decreasing in size. And this is how they move. Yes, there is a sideways, side muscles which allow it to move the foot itself, but the extension or decreasing the length of the foot is pure water. So whenever, for example, whenever the starfish latches on the muscle and try to pry it on, it's not just using the muscles, it's basically pulling it with the water. As a result, it can hold on tight for literally hours. And now our muscles can be contracted, or in the muscles of the mollusk can be contracted for an hour. So as a result, little by little, the clam or any other bivalves will slowly give out. And as soon as it will pry it open, the sea star will literally throw up its stomach inside of the shell. And then it will produce tons of digestive enzymes. Digestive enzymes will digest the mollusk inside of its own shell. Then the stomach will absorb it. Then the star will slurp the stomach back, and they'll move on. So in this particular case, feeding is really important. Those particular channels. So vascular system is extremely important for feeding as well as the gas exchange. They don't have a separate system, and then of course the locomotion. So here what we have, again, we're looking at the madreporites. From the madreporite, the madreporite goes into a stone channel, going to a ring channel, and then the water goes into the radial channels or canals, and then it will be attached to the ampulla. So water will be filling out the ampulla and the tube foot. Whenever the ampulla contracts, the tube foot, and here looking at the tube feet, so tube foot will increase in length. Say in this particular case, the tube foot is completely extended. Here it's not. Why? Because the ampulla is filled up with water, which is attached to that particular tube foot. So whenever it will contract, it will sprinkle over inside of the tube foot, so it will increase as well. So the structures within the starfish is relatively simple. I already told you have they have mouth, they have stomach, and then whatever is not digested will come out from the anus. So mouth, stomach, digestive system, and then the anus. Whenever they comes from the stomach, each of the arm will receive something which is known as a digestive gland. This is how the digestive nutrients will, or digested nutrients would be distributed to every single arm. Right under those particular digestive glands, most likely you will see something which is known as a gonads. Gonads, the testes as well as the ovaries will require a lot of nutrients in order to produce sperm and egg respectively. Therefore, they will be laying down right underneath. So whenever you're doing the actual dissection, the C-star, what you will do, you will cut the skin out and then literally gently lifted up that particular feather looking, and it's kind of usually greenish looking thing, and then you will see underneath of it will be the gonads. Because of the very simple structure of the C-star, if you cut the C-star in half, guess what happens? The C-star can regenerate. The half will reproduce, well, the two halves will reproduce or will repair the damage. As a result, eventually, given the time, you might ended up with two genetically identical, known as a fragmentation or regeneration. That would be asexual reproduction. Also, regarding the nervous system, they have a nerve ring. From the nerve ring, it will have the nerves going in each of the arms, because remember, all of those ampullas, contraction and relaxation have to be coordinated. They don't really have much of the sensors. They do have quite a few, like for example, they have an eye spot on the tip of each of the arms, but it's not really an eye. Which can they see? They can see, well, they can distinguish from bright light versus dark light, but it will not form images the one our eyes do. And those particular tube feet quite often will have a chemical receptors for taste/smell, because they live underwater. It's pretty much the same thing. So that's basically about the internal anatomy of the C-star. Make sure you understand how the water vascular system works, and let's kind of move into different groups. So evolved secondary, again, the larvae is bilateral, and then whenever it settles down, it will go into a 5-layer or 5-array symmetry. Okay. So here's the main groups. In our particular lab, we'll talk about you will see the examples of the C-stars. You will see the example of the brittle stars, the urchins, as well as the sand dollars. Sand dollars we have quite a few in Galveston Bay. We talk a little bit about the cilialis and then the sea cucumbers. Those ones considered Cyclidea, sea daisies, relatively small leaves, about yay big, lives in a deep ocean, I mean a really deep ocean, and they've been discovered, I would say, about 30-40 years ago. There is only 1 or 2 species are known, so we don't have those ones in the lab. But the rest of them you will be able to see, and you should be able to identify each one of those, because that particular questions will be on an upcoming lab practical, not the next week, but the week after. Also, I'll put the here next Friday, not tomorrow, the day, well, next Friday, whatever the date it is. Anyway, next Friday, not this one, but the Friday next. Let me real quick look it up. What's the actual date is. So next Friday I will have, so it will be 28th. October 28th, basically Friday before the lab practical. I will be in a lab, South Campus lab, in the first building, science building, lab 381. It's a bio 2 lab. So if you want to stop by, kind of take a look on the samples or anything like that, you are more than welcome to do so. So sea stars, the first group, one of the largest ones and the most recognizable ones. If you ever been in the ocean, like Cancun area, I love Yucatan Peninsula to begin with, really, really nice. Mexico, been there multiple times. Showing it, actually missing it. I was planning to go over the summer, but then the whole COVID situation, etc. So I didn't have the chance, but I really like it. And one of those particular things, that's the northern end of the Mesoamerican coral reef, and they have those particular creatures there. You can find them. They are pretty much harmless, but they're really nice to observe. Even if you're snorkeling, you can find all kinds of cuttlefish, and well, that would be a mollusk, but the sea stars and sea urchins and all that particular stuff. So have a radiating disc, and usually you'll have five arms. Sometimes it's 4, sometimes it's more than 5. Why? Because, well, for example, there was some sort of a predator which cut off one or eat off one of those particular arms, and there are 4 now. We have the sea star healing that particular part, but now it will be a 4-star sea star, so to speak, 4-arm sea star. What we're looking at the dorsal side of the sea star. Let me go to the next pictures. So here we go. That's a dorsal. It's a back side. That particular part right here, this off-center spot, this is the madrepori. The anus will be located almost at the center of the sea star. Now here you're looking at the multiple arm sea star known as the Podiuts, one of the largest sea stars we know of. It can grow about yay big, yes, about like 20 or so inches in diameter. That's a big sea stars. We don't have those nearby, to my knowledge, but Australia, if you go to Australia, Great Barrier Reef, you can find those ones there. So that's basically sea stars. I don't really have to go too much into that. We'll discuss that a little bit more at the lab, and also please watch the video. Now, brittle stars, looking very similar to sea stars. The only issue, or one of the main differences, that the arms will be extremely thin, much more flexible. So basically, you definitely, now you can clearly see the central disc and the long flexible arms. And the long flexible arms, basically the sea star, well, the brittle star will raise them up. It will be lined up with the tube feet, and they will try to capture whatever plankton they can. As soon as they capture it, they will move it down into the mouth. So mouth will be on the opposite side. That's again, and we're looking at the dorsal side. So brittle stars, and they usually, you can create the, well, you can see them in hundreds, if not thousands of them, all of them slowly waving their arms. Yes, they can move, but they generally, they sit on the bottom of the ocean on the substrate and waving their arms, capturing any kind of creatures, small creatures. Now, sea urchins as well as sand dollars. Sand dollars we have here is basically, think about the sea urchin as the sphere, and then it flattened out. As a matter of fact, I have some of the, this is the sand dollar essentially, and you can kind of see the 5-ray symmetry going in there, right? But in this particular case, it's not really flat. It's one of those particular sand dollars which were evolving, and that's another sand dollar. Again, we're talking about 5-ray symmetry, but it's flattened out. And nowadays, I don't think I have a modern sand dollar here. I need to get one. But it's nowadays are completely, well, pretty much flat, right? So sea urchins and sand dollars don't have arms, but they can have a row upon row upon row of the tube feet, and they are very important grazers in the in the Pacific coast. Pacific coast, one of the main algae there, proteus known as the brown kelp. Huge algae which are photosynthesizing, they're creating the forests of the brown kelp, and those particular forests sustain the life, literally like a regular forest. Those particular creatures feeding on the brown kelp, everything is fine, it's a kind of balance. There is a special predators known as the sea otters, which is feeding on the sea urchins, keeping them in check, until the humans came in and saying, hey, the fur of the otters is extremely nice, so we'll start hunting down the sea otters. And we almost hunted down them to the extinction. They are very protective species on the Pacific coast nowadays, and the species is coming back. It's a good story, but what it did, no natural enemies. The sea urchins start reproducing en masse, greatly reproduction. Now, what will happen? They are reproducing, they are grazing on those particular brown kelp forests, literally cutting them down. As soon as they are destroying those particular brown kelps, fields of the brown kelps, we ended up with losing the biodiversity. So now forests turn into, well, underwater forests, so to speak, turn into underwater desert. Now, once again, we are coming back, well, the sea otter is coming back. As a result, we are protecting those brown kelp forests. So quite interesting ecological connection between the brown kelp, the sea urchin, as well as the sea otters. Another one known as the sea lilies, also known as the crinoids. And those particular crinoids, they look like plants. As you can see, they sit on the bottom of the on a substrate attached to it, and they look kind of like lilies, hence the name sea lilies. And basically, this is the arm, and they waving the arms, and they are trying to capture whatever they can, whatever the plankton they can, and then digest it. Many of those particular sea lilies are attached, but they can, in many cases, they can remove, especially the sea feathers. Here we go. This is a sea feather. As you can see, it's attached. It's waving its arms. That's the highly modified arms to capture pretty much any kind of plankton. But if the plankton is not coming, the sea star can detach itself, not sea star, feather star, and then slowly float around, kind of settle down somewhere else and see if it will have a better spot, so to speak. So crawl around, and some of the crinoids can move around as well. Crinoids at some point of time, usually they live in a relatively shallow areas in ocean. And we have, in geology, we have a special limestone known as the Incrinal Limestone, which is basically infused with the parts of the crinoid. So at some point of time, about 200 or so millions of years ago, there was tons and tons of those particular crinoids in the tropical areas. They were dying out, getting buried, and then got into a sedimentary rock known as the Incrinal Limestone. Anyway, sea cucumbers, quite interesting kind of grazers. They basically lays on the again substrate, and they go through the sand, and they have a highly modified tube feed, which is literally shoving everything into their mouth, and then the whatever organic compounds will be digested, whatever is not will be expelled from the body. So very interesting creatures. In some areas they've been eaten. Galaturias, actually, I checked. You can we do eat sea stars in some areas. There is even sea star. We definitely eat Mediterranean as well as the Asian cuisine. That goes into the eat the sea urchins, and then Asian cuisine also dabble into the sea cucumbers. To my knowledge, I haven't tried the sea cucumbers yet, but I guess whenever I'm visiting like Korea and Japan, I probably will try it just out of the curiosity, so don't know how they taste. The sea urchins are okay. In many cases, they eating them fresh, I mean, raw. So that might be a little bit, but again, different people. So here's the last one, known as the sea daisies. Only 2 species known, has been discovered in mid-80s, and they are deep sea creatures. So we don't have to worry much about those ones. All right, so let's see. Okay, so next group, we'll talk about the chordates. So chordates is basically creatures which have something which known as a notochord. Again, shares the same embryonic development with the echinoderms, so Deutorostomes. And there is the actually 3 subphylum known as the, well, 2 of them are invertebrates and 1 of them is vertebrates. So here's what basically we looked before throughout our journey into the animal kingdom. We started with the sponges, asymmetrical parazoa, not really an animal per se, not plants, not fungi, very close to animals, but not animals yet, no symmetry. Then the first group of diploblastic, 2-layered animals, cnidarians, as well as networks. Anyway, so cnidarians and the comb jellies. So those ones, diploblastic layers on the ectoderm and the derm, no mesoderm, starting with the flatworms and down 3 creatures. Flatworms didn't have the body cavity, right? So asalamates. We didn't talk about the rotifers. However, we talked briefly about the brachiopods, not too much. And then we definitely discussed the mollusks and the annelids, and those ones were lophotrophozooans. Nematoda roundworms, those ones are actually the true coelomates. This is a pseudocoelomates, and they are combined with the group known as the art because what? Because of the exodus. They shed the outer skeleton. And here we're looking at the 2 major groups, echinoderms and the chordates. All right. So now we are on the chordates. Again, everything starts with the Cambrian period. Well, not everything, but everything starts showing up in the Cambrian period. That's why we start talking about Cambrian Explosion. And by the way, last week you're supposed to watch the video about the Cambrian Explosion, because periodically I'm getting the questions from the students who obviously didn't watch the video. So they're like, what kind of explosion? And I'm like, what do you mean? What was it, like a nuclear explosion? Was the asteroid hit the Earth? I'm like, no, it was explosion of life. Yeah, like that. But what exploded? I'm like, well, yeah, life. The number of creatures start showing up, multicellular. We start seeing tons and tons of different fossils. So in one of those particular group, vertebrates considered to be one of the most successful group of animals on the same, well, kind of in the same trend as the insects. Insects, though, stays a little bit smaller, or much smaller, I should say. However, they are in millions of species. The vertebrates, even though we consider to be one of the most successful groups of animals, in thousands. So, well, tens of thousands, as a matter of fact. So, backbone, and this is the backbone of the snake. As you can see, remember we're talking about the segmentation. You definitely can see the segments right here, repeating units over and over and over again. So chordates will have a structure known as a notochord. Okay, this is one of the very important characteristics of all of the chordates. Like I told you, me or you as an adult don't have a notochord no more, but we started, our embryo had it, and then the notochord was replaced by the backbone. And very important, please mark it down, dorsal hollow nerve cord. There might be a question regarding that on the test. Name the characteristics of the chordates. And I'll talk about that several times. So I'm just paraphrasing the question, but it most likely will be on a test as a short answer. name main characteristics of the chordates. Presence of the notochord, at least at the embryonic stage. Dorsal and hollow nerve cord. Remember where the nerve cord were in insects and arthropods in front. It was ventral. Now we're talking about the dorsal, right? So dorsal means on the back, and in our particular case, it's surrounded by the vertebral column. So once again, we are bilateral animals with deuterostomes. We are true xylomates. We do have xylom. So, and then there is two groups of invertebrates. They don't have the vertebrate column, known as the urochordates and cephalochordates. So cephalus, again, head. Those guys have a head. Those guys, not so much, at least as the adult one. And we'll discuss that momentarily. They're very close, well, close related to us from the genetic point of view, from the evolutionary point of view. So this is basically a possible phylogeny, how we are related to whom, so to speak. Ancestral deuterostomes, branched out, radial symmetry, bilateral symmetry. Radial symmetry, we ended up as adult and echinoderms. Then the presence of the notochord. So notochord right here. All of these creatures have the notochord. That's one of the reasons why I asked you to do the cladogram assignment, so you will know the derivative trait. So the presence of the notochord. Now, urochordata don't really have much going on as the adult. They are very exciting as the larvae though. Then we have the presence of brain. They sessile organisms, so they are as an adult they're losing the brain, no brain whatsoever. Again, next group successful but brainless. So presence of the brain means front end. Front end, we're looking at all of those creatures will have some sort of brain. And then we are, as soon as we started having the brain, head, brain, head. Which one is it first, chicken and egg, so to speak? Because as soon as we have a front end interacting with the environment, there will be a concentration. Lots of sensors will be on the front end. And then, of course, vertebral column. Why? To protect that particular vertebral, dorsal nerve cord. And then jaws and mineralized skeleton, further protection of the internal structure, including the nervous system. And now jaws. Why the jaw is important? Initially to grab the prey. Very, very important, because we are, or our ancestors are predators by nature. So in order to grab it and hold on and then digest it, have to have jaws. It's one of the very important characteristics. And the lung derivatives. We'll talk about how a specific group, specific fish known as the dipnoid double lung fishes are developed something which is similar to the lungs, and most likely this is the group with the specific lobe fins which gave us the terrestrial creatures, the legged creatures, starting with amphibians, which change into the primitive reptiles, and then the reptiles split into several groups. The modern reptiles, snakes, and the lizards, and the crocodiles, it's a different branch, and the tortoises is a different branch, and then the same branch we have the birds. And then another group of the primitive reptiles ended up with a slightly different strategy in life, and ended up being evolved into a modern mammals. So here we are on the brief evolution of the vertebrates. So phylum Chordata. So here we're looking at the hypothetical chordate. It has a very specific traits, which please make sure you know. They actually highlighted here for you. So A, have a notochord. It's a flexible rod. It's kind of like a cartilage rod goes. It's not really a cartilage, but cartilage like rod goes through the body of a particular creature. The whole basically body is suspended. It's a beginning of the internal skeleton. All right. Above the notochord, we have a dorsal hollow nerve cord. And remember, nerve system, nerve cord specifically, is developing from the ectoderm. So I think it's sure of that. I'll need to double check. I don't remember. But basically, what we have a special cells on the ectoderm kind of start moving in, and then they will create the tube, the nerve tube with the channel inside, hollow. That particular channel is filled up with the cerebral spinal fluid, and this is the brain as well as the nerve cord getting the nutrients, so to speak. So presence of the notochord, presence of the dorsal hollow nerve cord. Now pharyngeal slits. Initially they were not, well, they were predators, but they were sessile predators. They were sucking the water in, whatever the plankton they were able to get it would proceed into the digestive system, and then whatever is not digested will be coming out from anus, so complete digestive system. So those pharyngeal slits are important from the feeding point of view. And then this is how they were getting the oxygen gas exchange in the water. But later on, those particular pharyngeal slits, or gill slits if you wish, will start changing, turning, evolving into quite a few things. Jaws is one part of that. Then the brain, the very, very tender tissue needed addition. So many of those particular gill slits modified in a part of our skull. As a matter of fact, we, as each one of us, as the embryo, had those particular gill slits, and then they got modified gill arches, modified into many structures within our head, including these tiniest bones in our ear. We do possess some reminders of that particular gill slits. If you touch the tip of your ear, like right out, right there, you might feel tiny little bitty cartilage bumps on your ear. That's some reminders, whatever is left out of those particular gill slits. And no, we will not be able to grow the gills out of our ears or anything like that. But once again, it's extremely important from the overall development, because most of the structures, bone structures in our skull is actually derived from that part. So pharyngeal slits and post-anal tail. The tail is after the anus, and then of course there's a muscle segments and so on, and then we have a brain, again concentration of the nerve tissue at the front end. So we'll start looking at the cephalization, formation of the head. So again, same kind of the picture. So talking about dorsal nerve cord, the notochord, muscular segmented, of course, a segmented muscle, so muscle tail, which is a post-anal, meaning it goes after the anus, and then pharyngeal slits, and then we have the body cavity, right, where most of the internal organs located at, and so on. So that's the gesture of our ancestors about 700 or so millions of years ago. So the notochord, what it is? Longitudinal, so goes through the entire of the body, almost entire of the body, flexible rod, located between the digestive tube and the nerve cord. So nerve cord, notochord, digestive tube. Initial little support for a chordate, but again, in our particular case, we are substituting for actual bones. So it's much more complex and skeleton, and we have only remnants of embryonic notochord kind of in the middle of our vertebrate. We have a tiny little bit kind of dot looking structure, and that's the whatever is embryonic notochord used to be. All right, so again, important, the characteristic of all of the chordates: dorsal on the back, hollow nerve cord. And again, develops from the ectoderm, right? The outside layer, skin and the nerve tissue predominantly develops from that. It develops into a central nervous system, the spinal cord, and then the concentration of the nerves in the front end in the head will turn into a brain. Very, very important. And again, please remember that particular connection. Front end goes first into the environment, have to sense. Tons of sensors have to have a process units for all that information. And also respond, not only to process, but respond properly. Pharyngeal slits, gill slits, gills arches, extremely important. I'll show you some of the pictures. Don't have much time to spend on the comparative embryology or evolutionary embryology. Very interesting topics, but unfortunately tied up on time. As a matter of fact, chordates, just the chordates. In my undergrad, it was the second year, so 2 semesters just on the chordates. All right, and 2 semesters on vertebrates. My first year in undergraduate schools. So my freshman year, 2 semesters of invertebrate zoology and the 2 semesters of vertebrate zoology in the second, my sophomore year. So initially, suspension feeders, letting the water through, capturing whatever they can, but later on start to modify it for the gas exchange gills, right? And then they start modifying into a jaw, ear, neck, well, parts of the ear, as well as the head of the terrestrial vertebrates, well, the fishes. And then, speaking of which, well, I'll get there momentarily, but we'll talk about fish today, right? As the class, this is the largest class of the vertebrates, the fish. How to properly say. Think about me as a Russian professor who doesn't know how to say fish in plural. Is it fish or fishes? Please feel free to respond in the chat. So what do you think? Fish is a plural name for that, or fishes is a plural name for that? And yeah. Fishes. Well, maybe, I don't know. Say I'm a Russian professor. English is my second language. Or third. So please, anybody besides Jessica's paying attention to the class? What asking? No? I guess I'll mark everybody else out. Anyway, oh, more people are. So anyway, muscular postanal tail. I'll keep you in suspense a little bit. So muscular postanal tail, and again, we talk about cementation of the muscles. Very important propelling force for many aquatic species nowadays. We as humans don't have the tail, but most of the mammals do, right? Taxonomy. Again, we talk about the urochordates, the phallochordates, vertebrates. Majority of the lab 15, I think, the last lab for this, you will talk with the vertebrates, but I'll give you the example of the urochordates and cephalochordates as well. Tunicates, that's a urochordate. Tunicates is a very interesting group. They are marine suspension feeders. So they sit on the bottom of the ocean, they're sucking the water from one opening, they're letting the water from the another opening. So the interaction with the environment is minuscule because they don't have that particular active moving. They don't really have head, no brain. So they become very, very simple creatures as they adult. As the larvae, they are extremely interesting from the evolutionary point of view. Quite commonly referred to as the sea squirts, because this is what they do. They sit on the bottom of the ocean, they're taking in the sea, they're taking the water in and they're squirting it out. That's it. Not a very exciting life. but they are very exciting point of view, and I'll show it to you right here. So here we're looking at the larvae. Okay, larvae is very exciting because it shows us all of the characteristics, all of the main characteristics of the of the chordates. Have a dorsal hollow nerve cord, has a notochord, has a muscular tail, post-anal. They do have a brain or start something with a brain. It's free swimming. Everything is great. But that particular larvae, after a while swimming, is basically flips on the side and settles down. As soon as it settles down somewhere on the bottom of the ocean, it's losing most of its advanced features, so to speak, or chordate features, and it turns into a ciscord, literally. Here we go. So see, that's why it's called tunicate, because it's kind of look like almost like a coral, or almost like the anemone. As the incurrent siphon, the water goes in. We still have some of the gills which are taking the water with oxygen and the nutrients. Any kind of plankton goes there, and the water comes out. Whatever is concentrating going into the digestive system, and whatever is not digested comes out with the excurrent or through water. Filtering food particles, so no brain, no head, well, no head / no brain, not much of the advances or advanced features remains. But again, as the larvae, pure chordate right there. But over a period of time, just simple goes into a simple life. No worries about pretty much anything. You're sitting on the bottom of the ocean, sucking it in, almost like a vegetative state, right? Almost. It's not, it's an animal. It's not by any means it's plant or anything like that. But in this particular case, very, very unexciting life, at least from our perspective, even though sometimes I really want to feel like a ciscord, you know, sit somewhere on the side of the ocean and do absolutely nothing. But that's beside the point. Sometimes you need to take a break. Now, next group, Cephalochordata. So Cephalochordata, this particular creature known as a lancelet, because it looks like a cutting instrument of the medieval medicine, which was lancelet, blade like. Those guys are staying as the chordates or keeping those particular traits as the chordates as the adults. One of the ideas, again, saying that maybe at some point of time, due to some sort of mutation, the mistaken DNA, that particular larvae, instead of settling down and turning into a tunicate, actually start free living, did not change, and end up being this guy. As the adult, continue to have the nerve cord, the notochord, the tube. Now it has tentacles, so it's moving in, moving some water in. Continue to have pharyngeal slits. Whatever is going in, it usually stays buried in the sand. So not extremely, again, exciting life either on the modern of the cephalochordate. But that was a very important step. Now, as the adult, having all of those and the tail, it has a tail, it can propel, it can gain speed, it can start capturing the slower moving prey. But in this particular case, think about it. In order to sense the prey, what you have happened? You have to increase the amount of sensors on the front end. Yeah, as soon as you start increasing the amount of sensors on the front end, chemical sensors, pressure sensors, electrical sensors, the light sensors, you increasing the brain, and you will end up as the fish. Fish were a first, well, besides they were a first group of vertebrates, they were brainiacs. They had a huge brain comparatively to the other creatures. Why? Too many sensors. You have way too much information, need to be processed as quickly as possible. All right. So this one don't have much of the brain. It starts having some, but because it's most of its time kind of sitting in the bottom, buried, not very exciting. Again, not very exciting, but from the perspective of the evolution, extremely important part. Yeah. And you will see those particular, they are about 2 centimeters, 2.5 centimeters each. So they are about 2.5, 3 inches. So lanceolate suspension feeders, small segmented fish-like, exhibit all of the chordates characteristics. Later on, you will see the model. I'll show you the model in the lab next week. So, but once again, have exactly all of the characteristics, but now not only as the larvae, but also as the adult. All right, that's important part. Now, the current life history of the tunicates, once again, probably were first to evolve based on the genomic structure of that. And once again, we think that might that the tunicates, specifically the tunicate larvae, start reproducing somehow on its own due to some sort of mutation, possible. All right. Cephalochordata, on another hand, and vertebrata are very close related from the genetic point of view and the structures, of course. Now, let's talk a little bit about Hox genes. Anybody knows about the Hox genes? You're supposed to talk about that during the Bio 1. If you took Bio 1, you should, your professor should be, or your instructor should talk to you about the Hox genes. Anybody heard about the Hox genes? Do you know, do I need to tell about those exciting genes which we discovered? Hox, yes, no, maybe. Do you know about Hox genes, Hox 3, or you don't remember? All right, okay, cool. At least one student were able to say something about that. Now, let's see, Hox genes. There is, you know, genes is segments of the DNA which are responsible for a particular trait, right? That's the typical gene. We discovered a group of genes which are responsible for developing, well, they kind of starting, they are kicking off the development of the particular organs or group of organs. All right, makes sense? So let me give you a quick example. This is what, whenever I'm teaching Bio 1, telling to my students, there is a Hox gene which is responsible for developing of an eye, or eyes in plural. So far, so good. So there is a specific gene known as the Hox genes for the eye. You don't have that particular gene, there will be no eyes developing, because basically the organism, whenever it's developing, does not read, even though all of the genes for the what color of the eye, etc., what type of the eye might be there, because there is no Hox genes, it's not initiating the developing of that particular organ. And the scientists played with the fruit flies. They were able to find in the embryo and cut out from the DNA that particular Hox genes which were responsible for developing of the fruit fly eyes. So far, so good. So that particular embryo developed into a fruit fly. All good, nice looking fruit fly, but that particular fruit fly didn't have eyes. It was completely eyeless, because once again, the Hox genes which supposed to initiate the developing of the eye was absent. So the scientists run several generations of those particular completely eyeless creatures, and basically they were making sure that nothing will interfere with the absence of the Hox genes. Neither of the other genes will take care of the formation of the eyes. So all of those generations of the fruit flies were completely eyeless. Again, the organism, the embryo didn't have the instructions saying, hey, you start developing your eye. And then the embryo saying, oh, okay, I am a fruit fly, so this is the eyes I need to, this is how, what kind of eyes I need to develop. All good. Any questions, any problems on that? All right. Now, next step of that particular experiment was to take a Hox gene from a different animal. As a matter of fact, they used the flounder fish. Flounder fish, Hox genes, which is responsible for developing of the eyes in a fish. We're talking about insects, protostome, arthropod, versus fish, vertebrate, Deutorostome. So they taking the Hox gene from the fish, they inserting in a developing embryo of the fruit fly and waiting for a miracle to show up, and the miracle indeed happened. What kind of miracle happened? The next generation, that particular embryo with the Hox genes from a flounder developed eyes. What kind of eyes? Was it the developing of the eyes of the flounder, or was it the eyes of the insect? It was the eyes of the insect, because now that particular embryo has the instruction, start developing the eyes. Just that particular line, develop the eyes. And then the embryo knows what kind of eyes it's supposed to have. So Hox genes are the instruction in developing of a particular organs. All right. So simplified speaking, it's not as straightforward as it is, but we're not in genetics. So let's go back to our creatures. Now let's take a look. Nerve cord of the lancelet embryo. Responsible for the nerve cord, there is several genes which are responsible developing the parts of the nerve cord, BF1, OTX, Hox3. The same genes are responsible for developing our own brains. As a matter of fact, BF and OTX are responsible for developing the front brain and the midbrain. Hox3 is responsible for developing of hindbrain. You're cutting that one out, the hindbrain is not developing. Hindbrain controls most of our automative responses. You don't think about heart beating, right? Like, oh, I need to contract my left atrium, right atrium, etc. You don't think about it. That's an automative response. Hindbrain. Think about breathing until I tell you so. And yes, you can override if you want. I'm not breathing for some time. Can you kill yourself without just stopping your breathing? No, you pass out, and then your automative organism will start and continue to breathe. All right. So those particular Hox genes in the lancelet and in vertebrate are identical, meaning that we had the common ancestor. And yeah, Hox genes are quite interesting. So here we go. Let's take a look on those particular chordate ancestors. Probably a tunicate larvae, maybe not, but with that. And also, well, the chordate ancestor, tunicates evolved first, and then the chordate larvae created the cephalochordates, and then there is a group of jawless fish, or fishes. So fish or fishes, some people say fish, some people are saying fishes, and we'll get there momentarily. So in this particular case, they don't have jaws just yet. They haven't created, they have teeth, no jaws. Jaws are one of the very, very important evolutionary steps in the evolution of the vertebrates, including ours. We have jaws, upper and lower, and so on. All right, so phylum chordates, overall 7 classes. 7 classes, extant classes, means the one which is present right now. We have 7 of them. 3 of them called fishes. If you answered fish, according to the the English rules or the English language, you can either say for the plural, you can either say fishes or fish. It will be acceptable. Either or will do. Okay, so well, all of you were correct when you were saying fishes or fish, doesn't matter. It was okay with like one fish, okay, many fish is okay. Many fishes is okay as well. One fishes is not okay. Make sense? All right. So the three first classes known as a fish, fishes, and the other ones, I had that particular conversation with one of my students a couple of years ago. She's like, whenever you're saying fishes, it doesn't sound good. I'm like, I don't know. That's the English. That's don't kill the messenger specifically. But in this particular case, four other known as the tetrapods. And what's really important that particular group fish, or specific group of the fish, give us the creatures which has four-legged creatures. Well, we technically don't have four, we have only 2 feet and then 2 arms, but essentially that's the all evolution of the humans. We already talked about the evolution of the front end, the bipedal, right? Why the bipedalism occurred, the saving of energy, looking further, and so on and so forth. But essentially, we belong to that particular group as well. And then, of course, we have segmented vertebrates, and we'll see that later on, 2 weeks, 3 weeks from now, when we'll start talking about the bone structures and tissues, etc. So, shared characteristics of pretty much all of the vertebrates. Vertebral column. We have a special protection for our spinal cord. Cranium. Cranium, the part which is protecting the brain. The pronounced cephalization. We do have all of the vertebrates have the front end with the concentration of the sensors, with the high concentration of the nerve tissue, and quite often with the protection of that particular tissue, brain, so to speak. Muscles are attached to the endoskeleton, unlike arthropods, where the muscles are attached to the exoskeleton. So, origin of the head. Again, very different head. Well, the origin of our head is very different than the origin of, let's say, octopus head. Yet the same idea. Front end goes into the environment, or interacting with the environment. Increase of sensors, increase of the nerve tissues. Ta-da! And now we start looking at the active predation. Remember, I told you that particular sand-buried lancelet don't really have much of the brain, but as soon as it left the sand and start actively something slow, about 700 to 1 billion of years ago, this is where we start seeing the increase of the sensors. Increase of the sensors, or need to increase the number of sensors to sense that particular prey. As soon as you need to sense the prey, you have to have a bigger brain. As a result, fish will become first brainiacs of that particular animal group. Very, very important. But again, the presence of brain is slightly exaggerated, or need for the brain. Starfish survived for millions of years, brainless. Cnidarians, the jellyfish, completely brainless, extremely successful group. So brain, yes, for some of those, for some of the groups, it's extremely important. For others, like, I'm brainless, I'm happy about that. So in this particular case, so again, as soon as we start seeing eyes and other sensory organs, we start seeing increase in brains. And we'll talk about that later when we'll discuss the evolution of the us, mammals. Well, the humans we already discussed in the beginning, but mammals, why the mammals comparatively to the other group has the biggest brains. And I have theory about that. So this is what happens whenever we start getting the dorsal nerve cord. Remember the notochord, and then we have the ectoderm, and some of the cells, known as the neural cells, they will start pushing part of the ectoderm inside, creating a hollow cord, hollow nerve cord. Okay, very interesting how the developing of the nervous tissue in the vertebrates. This is how our spinal cord will have a channel. Cerebral spinal fluid is circulating and then it goes into the ventricles of the brain and so on. We'll talk a little bit about that during the anatomy. Nervous tissue closer to the end of the semester. So neural crest cell, besides the pushing the ectoderm inside, also gives quite a few structures on the skull, and that's of course the baby skull. You can see those ones. So now craniates with the skull, brain of course, but with the skull, evolved somewhere around 550, maybe a little bit 600 millions of years ago. So again, Cambrian explosion, at least 530 millions of years ago, because we have a distinct fossils for that. Those particular fossils are quite strange looking, and this is one of the creatures known as the Echinoderms. And that one, again, look, you clearly see even in the 500 plus millions of years old fossil, you can see the segments of muscles, right? And in this case, clearly see the front end. And if you're looking under a dissecting scope, you start seeing the other structures. But that particular creature is about an inch long, 3 centimeters, a little bit like inch and a quarter. So not a very long, not a very large creature. However, this is the first one which start, well, one of the first ones which start actively hunting for slow or prey. This guy, Phaeaconchitus, definitely had a skull, much larger creature. Well, not much larger, but much more vicious, let's put it this way, because it was more advanced. It had a bigger brain. We presume it had a much bigger amount of sensors, so was able to find that prey easier. So within the fish, we have several groups. Ostracoderms, like extinct. Then we have Agnatus. Gnatus, known as the jaw. Agnatus means jawless. And within the jawless, we have two groups known as the hagfish, and we'll talk about those. Also craniates, and here we're talking about the vertebrates. Hagfishes are invertebrate because they don't have a complete vertebral column. We'll talk about that. It's on the iffy side. So Agnatus, jawless fish. Again, fish and lamprey, paired appendages are not present. Remember, most of the fish will have a paired appendages. We'll talk about that. They don't have a bony skeleton. They have a skeleton. It does not mean that they don't have a skeleton, but the skeleton is made out of cartilage. No jaws, no jaws whatsoever. As a matter of fact, hagfish, if you pull it out, we have those in Galveston Bay. I'm not a big fisherman, just periodically doing it with friends. However, whenever you're looking at that, and mine a couple of years ago pulled out one of those hagfishes, and I'm like, "Oh, hagfish!" He's like, "Hey, it's a really nasty looking thing. It looks like a worm. I mean, large. It's. I'll show you the pictures. I think that the very next one. So, circular sucking mouth part. Some of them, specifically lampreys, have no jaws just yet. Here we go. This is a hagfish. Look at this. Look like literally like a worm. It's slimy. It has a slimy gland, but the mouth and then the sensors important. So the least evolved creatures, yet they survive for quite a few years, for millions of years, as a matter of fact. So again, looking very similar to a large worm, and apparently doesn't taste good. So Hagfishes will have a skull, but made out of cartilage, axial rod of cartilage, which is derived from the notch. They don't really have a vertebrate, so technically they are invertebrates. And the Cambrian period, again, that particular Cambrian explosion, all that particular exciting time for all of the paleontologists / zoologists or biologists of that particular direction. But in this case, we are very, very important that during the Cambrian explosion, out of 45, 47 on phylum, we can trace down about 35 to 37 modern phylum back to the Cambrian. That's why it's called explosion of life. So vertebrates have the vertebrate enclosing the spinal cord, and then the, of course, protection of the brain, elaborate skull, fin rays in aquatic forms, and then moving into the feet, so to speak. Lampreys, cephalodismorphy, and in this particular case, all this of the vertebrates or living lineages, they are still survive. They are quite common if you'll go into your Great Lakes, for example. For one or another reason, we brought those creatures there, and they are reproducing en masse because they don't have the natural predators in the Great Lakes. Hence the and they parasitizing on the other fish. So yeah, have a cartilage segments surrounding notochord. In some cases, they might even arching partly. They're not fully enclosing the nerve cord. So a very simple group of fish, and once again, no jaws. So here we're looking at the lamprey, and as you can see, already have an eye, all of those gill slits, and this is the mouth, sucking part, and tons of teeth, very sharp. And what it does, essentially, see, lots of teeth. And what it does is basically finding the prey, and it's cutting with the teeth through the skin, and then it's feeding off the blood. It's literally feeding the blood. Periodically, it can detach itself. There will be a scar on the carp. Well, that's a freshwater carp, of course, but also can be on the salmon or anything like that. So yeah, suction carp, suction carp, they will be drinking blood. Is it possible for that to get on human, like a leech? I'm like, I don't really heard any exact, any recorded cases of that, because with those particular teeth, when it will try to bite you, you definitely will feel it and you'll shrug it off. Besides, it's a fairly large fish, big, so you definitely will see that something is trying to attach to your leg or whatever. So yeah, there was a very interesting group, completely extinct nowadays, known as the conodonts. And this is how the conodonts presumably look. They were a tiny little bitty one, and they had mouths filled with the teeth, very strong, very, very sharp. And how do we know about those particular conodonts? These structures or the fossils, we almost don't have those particular conodonts as the fossils, but we have tons of their teeth all over the places in most of the, whenever we're looking at the particular layers of sedimentary rocks, you definitely will find those particular teeth, tons of them. They were very, very abundant a long time ago. So another group of the fish, jawless fish at that time, start going for armor. They start protecting what we. So defensive plates of bone on top of the skin. And we still have some of the modern fish which doing that, but it's just the similar idea how to protect the body. It's not, they're not related or close related to each other, but once again, we start seeing the eyes, the eye forming images, and then they have tons of different sensors. So the brain is increasing. Again, fish, comparatively to the other groups of animals at that time, were really, really smart, so to speak, or not necessarily smart, but had a large brains. So mineralization, very important part when this is where the cartilage will be substituted for the bone, right? And the vertebrate endoskeleton will become fully mineralized. But if you think about it, like human babies, whenever they're born, many of the structures, including like here on top of the head, they have made out of cartilage. As a matter of fact, that particular part is extremely important because whenever during the birth the skull kind of collapsing, it's becoming smaller, and so it can go through the birth canal, and then it will open up, and over a period of a couple of years, that part right there on top, the cartilage part, will mineralize and become skull. And skull is one of the hardest or strongest bones which we know. The front end right here is one of the strongest bones we know of to protect the brain, right? So now Notostomes. Notostomes are fish with the jaw. Jawless, of course, vertebrates are far outnumbered because jaw was a very important evolutionary advantage. Not only to grab to the or to cut the prey with the teeth, but also to grab it and hold on to it and then slowly devour on that part. All right, so here what we're looking at, very important. Remember I talked about the gill arches. This is how it was working. We have a comparative anatomy, comparative embryology anatomy, very interesting parts of cranium, the part which were of skull, which were protecting the brain. And then we start seeing some of those particular gill arches changing in order to have that grab of what? Of the prey. Our ancestors were vicious predators, extremely vicious predators. So in this particular case, some of them ended up being the jaws, as you can see that those particular gill arches changing to upper lower jaw, and then this the red part added to the strength of the jaw, because well, as the jaw is growing, the prey start modifying as well, start getting bigger, stronger, so you have to be a strong jaw with the very large teeth. And this is what we are kind of talk a little bit about some of the armored placoderms. They already have jaws, paired appendages, everything is fine. And look at this particular creature. Yeah, so we start looking at the teeth, and this is the representation of the placoderm, 10, 15, 20 feet long. Some of them, some of them were very, very formidable predators of those seas during the Devonian era. We're talking about 400-ish years ago. And then there was another group of start protecting the skull, at least, from any kind of predators. And then there was another really interesting group known as the osteoichthyans. Osteo means, you probably heard about osteoporosis. Osteoporosis, loss of bone density. Osteos means bone. Ichthyos means ichthyar, right? Ichthyos means fish, bony fish. During the Devonian era, we ended up with a very interesting group of fish which didn't use those very heavy plates, armor plates on their body. As a result, they become much faster, much more faster, and they ended up having the upper hand. So those guys kind of got destroyed by the initially smaller, and later on we ended up with a bigger fish, which is known nowadays as the bony fish. Majority of bony fish, I don't think I have somewhere, I think in the office. I have the imprint of the fish, quite nice one, but I'll bring it next time probably. So In this particular case, evolution of jawfish and the amphibians. I know I'm almost out of the time. Actually, I'm out of time. Give me just a moment. I'll talk about, actually, no, it's right here. Let's stop right here. We'll stop with the on the sharks. We'll start next time, second part of that lecture from the sharks.